but how you can actually use those models to interpret experimental data, cardiac optical imaging data in particular, and how this can help in designing new optical imaging technologies. All right, so we saw a couple of movies from optical imaging in the previous talk. So if you're not familiar with optical imaging, basically this is schematics of a fairly simple epifluorescence setup where you have a perfused preparation of tissue, a whole heart or a coronary perfused ventricular preparation, which is stained with a voltage-sensitive dye, and upon excitation at the appropriate wavelength, this dye will fluoresce in a voltage-sensitive manner, and this fluorescence can then be detected using CCD or photodiode arrays. Now, optical imaging, as any experimental tool, has some limitations, but it offers some great advantages as well. So it can allow for quite high spatiotemporal resolution, and also offers the possibility to do multimodal imaging, so you could simultaneously acquire voltage and calcium, for example. Now, as we know, and as we saw in previous talk as well, electrical wave propagation in the heart is essentially three-dimensional. And so one of the questions that we've been trying to address in the last couple of years is whether we can use optical imaging to actually gain three-dimensional information about cardiac electrical activity. So I'll start with some background. This is effectively recapitulating my previous schematics, where in epifluorescence imaging you have excitation light shown onto the epicardium and then fluorescence collected from the epicardium. However, the actual, what is really going on is slightly more complicated, because photons do penetrate into tissue where they get scattered before they actually get absorbed by the dye. And fluorescence will follow a similarly quite random pattern because of scattering into the tissue. And as a result of that, what you're actually measuring on the surface in epifluorescence imaging is not simply the surface signal, but is really a subsurface average, is an optical average of electrical activity in a quite large subsurface volume. This is using a biophotonics model where we have actually calculated the contribution of each subsurface voxel to a single pixel on the surface, in this case here. And you can see that in the case of dye 4-NEPS, which is a widely used dye, you can actually get contributions from quite deep into the tissue, so up to 1 millimeter, and also quite far away from where you're actually looking at. So although you could argue this is a limitation of optical imaging, Chris Hyatt a couple of years ago actually found a way to exploit this property of cardiac optical signals, as he found that the shape of the optical upstrokes, that's the depolarizing phase in the optical signal, very strongly correlate to the actual wave orientation, the subsurface average of the wavefront orientation. So where he basically showed, using computational model initially, that the location of the maximal time derivative in the signal strongly correlates to the subsurface wavefront angle with respect to the epicardial surface. And since then, this has been verified experimentally in various tissue preparations. Now, as I said, computational modeling has been quite important in the interpretation of cardiac optical signals. So how do we go about modeling cardiac optical signals? Well, usually we start with an electrophysiological model. It can be a monodomain, bidomain model, just basically a model that gives you membrane potential in space and time. We also need a biophotonics model. So this is the model that will tell you how light propagates into the tissue. So you can, under some approximations, you can use a simple diffusion equation, or if you want to be more accurate, you use a Monte Carlo code, or you try to solve the radiative transfer equation straight away. So basically, this will tell you how excitation light propagates, gets scattered in tissue, and how fluorescence is actually scattered back out of tissue, and how you can detect it, basically. And so then you combine those two under the assumptions, first of all, that a voltage-sensitive dye responds linearly to a change in transmembrane potential, which is a reasonably valid assumption. And basically, what you do is you perform a spatial integration, so a convolution of your 3D distribution of membrane potential in space with an optical weight of each voxel. And this optical weight is effectively dependent on the excitation light that reaches that voxel and the unit point spread function that voxel contributes in terms of fluorescence onto the surface. So you can formulate this as a linear problem where you have a transfer matrix that transforms your membrane potential into a surface cardiac optical signal. This modeling approach has been used quite successfully by various groups to actually interpret cardiac optical signals. But we actually now are trying to go one step further, is given a set of experimental recordings, can we actually recover the underlying electrical activity? So in other words, can we solve the optical inverse problem? Now this problem, as in many cases in biomedical imaging, is an ill-posed problem. So it can suffer from many things. If you don't have enough data, this problem can be highly underdetermined. So you have too many degrees of freedom, you cannot find a unique solution. So this, to take care of this, you really need to be careful about the design of your optical setup. But even if you would have no or very little degrees of freedom, you still have to be careful because this transfer matrix can contain small eigenvalues, which basically will result in very large distortions due to noise in your 3D reconstruction. And here we haven't invented anything, we have just used standard regularization schemes to solve the inverse problem that have been around for quite a while. And this is just one of the schemes that you can use, which is called the Tikhonov regularization, and which for a good choice of this parameter, regularization parameter lambda, gives you basically the best reconstruction possible in terms of a trade-off between over-smoothing your solution but being far away from what you're trying to actually reconstruct, or maybe not over-smoothing the solution but then having an image that is completely distorted by noise. So we've actually moved on to solve this inverse problem using a system that provides enough data to actually have a more or less, or a less ill-posed problem than you would, for example, have with just conventional epifluorescence imaging. And this is a setup that was developed by Elizabeth Hillman at Columbia University. She's a neuroimaging person, so she has developed this system and validated for brain hemodynamics. But we convinced her that heart is also interesting. And so we modified the setup, adding a Langendorf perfusion apparatus so we did experiments with a rat heart using dye-4 NEPs. And the way the system works is effectively scanning the surface with an incident beam of excitation light. So this is the scan pattern, so these are line scans. And for each position of the beam, we actually collect light at various distances from this incident beam. Using appropriate optics and photodiodes. And so the underlying idea, although it's a huge simplification, but the underlying idea is that if you have a large, or if you have a small source detector separation, as illustrated here, you're actually probing in the vicinity of the epicardial surface, so you're not probing very deep into the tissue. Whereas if you have a large source detector separation, then the signal must contain contribution from much deeper into the tissue. And what the inverse solution actually, or the inversion of this transfer matrix tries to achieve is to extract this information for the different transmural layers. So this is just one example of the reconstructions that we achieved. So obviously we first tested the technique using models. So this was in rat. So these are time-space plots where the space component is the depth. And so we were testing two cases, effectively pacing the epicardium, pacing the endocardium, and looking whether we can actually see propagation from epi to endo when we pace epi and propagation from endo to epi when we pace endo, which seems quite trivial, but it's not guaranteed when you solve the inverse problem. So this was the first thing we started with, and indeed we were able to actually reconstruct at quite high spatial resolution the transmural propagation, both for epicardial and endocardial pacing. So this is the resolution that we achieve with this system, spatial resolution, 0.37 in plane millimeter and 0.2 millimeter in depth. Now this is the in-plane lateral resolution is basically determined by your scanning system, by your scan steps, whereas the in-depth resolution is determined by the dye that you're using, so how deep can you actually penetrate into tissue, and how many source detector separations that you're using. Now, laminar optical tomography really serves as some sort of proof of principle, but it still suffers quite major limitations. One of them is maximal depth probe is of about 1.5 to 2 millimeter. That's probably fine for a rat heart, but we can't use this technique to map all the way through a left ventricle of a pig, for example. And although those line scans are actually fairly fast in terms of the technology, they're still too slow for what we are trying to achieve, because cardiac dynamics are very rapid. And so the effective temporal resolution of 0.5 millisecond that we had in the reconstruction, we could only achieve this through gating. So we had to actually average over many, many beats where the system was gated with the stimulus. So the most recent work has actually been trying to address those 2 points, so increase depth penetration and achieve rapid acquisition. And this is just the final slide. This is our current ongoing work. So in terms of probing deeper into tissue, we have now access to a new class of near-infrared voltage-sensitive dyes. So this just shows penetration of light at two different wavelengths, 532 nanometer and 660 nanometer, into a prep of pig left ventricular tissue, where you can clearly see that the green light, which is the excitation light of dye 4, drops off very rapidly, whereas red light, the excitation light of our new dyes, penetrates much deeper into the tissue. And then in terms of probing faster, we actually now we have built this kind of system where when we have a wedge preparation, we can simultaneously image endocardium and epicardium. So we have always a recording in epifluorescence mode and one in transillumination mode, and we can also quite rapidly alternate illumination site from epi back to endo and so on. And this provides enough data to have a well, or more or less well-posed inverse solution, which has been assessed at this stage using computational models and optical phantoms, and this is a kind of reconstructions that we can achieve. So to conclude, well, we've shown that high-resolution depth-resolve optical imaging in principle is feasible in cardiac tissue. Multiphysics modeling, so combining biophotonics and electrophysiological models, play a very important role in this approach. Current work is the experimental validation of this 3D reconstruction technique. So this is the setup that we now have built at Leeds, where we achieved the fast switch of illumination using electronically triggered LED arrays. And further work will focus on actually improving the accuracy of our reconstruction by adding additional constraint onto the reconstruction. So by adding electrical recordings and also performing anatomically accurate reconstruction using DT-MRI-based modeling. And so finally, I'd like to thank well, people of my lab, collaboration with Aaron Holden and Alan Benson in terms of the DT-MRI modeling, long-standing collaboration with Arkady Pertsov. Thank you.
Your photon diffusion model describes correctly how the light penetrates into the tissue, but it's missing the other half of the problem.
Yeah, I agree.
And so you can't really use that until you resolve the other problem, because your lens effects is going to change the distribution. And so every time you do an experiment, you must define your numerical aperture and your magnification conditions. Otherwise, you cannot predict anything.
Yeah, I totally agree. And actually, more recently, we're using Monte Carlo, only Monte Carlo-based simulations. Monte Carlo.
Okay.
So, and actually, for the laminar optical tomography, we do take into account all the effects of the optics onto the 3D reconstruction.
Right. So the problem I have with the experiments that you showed, showing beautiful anisotropy of propagation. You can only do that if you have the correct numerical aperture. You have to have a very large numerical aperture and very narrow depth of field to see it. Otherwise, it blends in and becomes circular. And Igor and I published that a number of years ago in JCE.
But the LUT is not using like a wide, it's more like a microscopical approach. So we can discuss about the technical details of the technique, but yeah.
Okay, we should probably move on.